afternoon and welcome all to my presentation, which is about genomic selection in pigs and poultry, and in particular about the potential of using FAANG resources and other omics data to further improve genomic selection. I would like to start with making a few acknowledgements, in particular the Gene Switch project, which has been funded by the European Union. And in there, I'm heavily collaborating within Work Package 4, which is about development of new genomic prediction models. And there the logos of the partners involved are listed here. In particular, I want to mention Bruno Perez and Marco Bink from Hendrix Genetics. I'll be presenting a few results from Bruno during my presentation. And some of this work is based on a collaboration with the Jackson Laboratory in the US that have provided us with data from mice. So in modern pig and poultry breeding programs, genomic prediction really is the core engine. That's where the whole thing, the breeding program is sort of the heart of the breeding program is genomic prediction. And the input to that is, as you're probably well all aware of, is that genotypes, phenotypes, and pedigree of animals. In these modern breeding programs, sizable reference populations are available, and this, depending on the trait, these may comprise of a few thousand animals up to hundreds of thousands of animals. And these reference populations have been built up across generations in the last decade or a bit longer. So one thing that these breeding programs tend to do is keep on expanding their breeding goals, meaning adding more traits and trying to put more emphasis on some of these traits. So when doing this, an important challenge is to maintain or to increase the rate of genetic gain for each of those traits. So from a breeding program perspective, you would always be looking to be able to apply more selection pressure. The more selection pressure you can apply, this enables you to redirect it either to different traits or, for instance, to say, okay, if we can apply more selection pressure, we can also relax that and be more stringent on genetic diversity, that we try and avoid losing that. So the big question, of course, is how to achieve this? So for that, it's useful to have a brief look at the breeder's equation to come and realize how do we realize the impact of genomic selection relative to traditional selection. And what traditional selection is really depends on the trait and the species you're looking at. So here I made a sort of conceptual table for 4 of the main species in livestock: pigs, layers, broilers, and dairy cattle. And I tried to line out where I think, for the majority of the traits in those species, the benefit of genomic selection comes in. So whether it's changing I, so the selection intensity, it's changing R, so the accuracy of selection, or changing L, it's decreasing the generation interval. And as you can see from this table here, it varies a bit across species. But it seems that in all cases, it's definitely beneficial to try and improve selection accuracy. So looking at the breeder's equations, the different components, there's very little we can do about sigma 8, the genetic standard deviation. The other components, the selection intensity and the generation interval, they are mostly determined by the breeding program design, whereas the accuracy is actually mostly is partly also determined by breeding program design, but this is the parameter that is most heavily affected by genomic prediction and the reference population characteristics. So this expected accuracy of genomic prediction, well, there's been a number of formulae developed over time. The Detweiler equation was the first one, and I just provide one representation of this, which says that the accuracy is dependent on the size of the reference population, So bigger reference populations means higher accuracy. Higher heritability also means higher accuracy. And then we have this population parameter that has been evolved over time, and now we tend to term it ME, the effective number of chromosome segments. And if we term that a population parameter, it seems to, in the context of this formula, seems to suggest, okay, it's a fixed thing. There's not much we can do about it. So I think in most applications, that's probably also the view that has been taken, is that this ME, this population parameter, as soon as we have sufficiently high SNP density on the panels used for genomic prediction, that's then we probably have at least 90% of the job done. So the idea is that if you have sufficiently high SNP density, the level of LD between SNPs and QTL is good enough for your predictions. And because of the limited effective population size in livestock species, typically within breeds or lines, 50K SNP chips provide sufficiently high density for this. But then having said that, how can we then try and increase the selection accuracy? Is there really nothing much we can do about it? So far, the common approach has been to increase the size of the reference population, but that is subject to diminishing returns. And at some stage, you've reached your testing capacity, and you've gone back in time and genotype all the animals that you've ever phenotyped. So what else is there to do? So one line of research is to try and enrich the SNP panel for loci with strong association with the traits of interest. And a lot of work has been done on GWAS using whole genome sequence data, and then try and find SNPs with strong association, put these on the 50K SNP chips. And a similar approach is to try and use biological. So, for instance, FAANG information to prioritize loci starting from whole genome sequence data. Another line that is being followed, and there's other presentations in this conference about this for sure, is to use additional omics data. So not just genomics data, not just SNP data, but for instance, using microbiomic data, gene expression data, methylation data, etc. And this hopefully gives us a better handle on prediction accuracy, but it should definitely also be useful to get a better handle on phenotypic variance. So how do we then go about doing this? So if we want to use other omics data, that's the idea is that we could measure those on the individual animals. So selection candidates or part of the animals in the reference population, on those we could measure on the individual level additional omics data and use additional explanatory variables in the model. And this can then be used to better predict phenotypes that could subsequently be used for genomic prediction. So the FAANG data is more a measure at the population level. It doesn't give additional data or additional observations for the individual animals in genomic prediction models, but it gives a better view at the population level. And this allows to prioritize or to group genomic regions that should be, well, let's say, boosted in the genomic prediction models. And then we're not so dependent on the linkage disequilibrium between SNPs and causal loci to actually try and find those. So in the gene switch project, we actually try to consider both these approaches. So just to say a little bit more about the rationale of the genomic prediction development that we're taking in gene switch. So the traditional source of information are genotypes and phenotypes. We want to top this up with functional information. And to this end, we are developing both Bayesian and machine learning models that can consider all these this traditional, but also this new functional information simultaneously. And then the project deals both with chicken and pigs. And for chicken, we are additionally generating a fine map QTL that we can use in the models. And for the pigs, we are additionally trying to find eQTLs that can be used in the genomic prediction models. So combining all these things together, towards the end of the project, we will validate these new models using these new types of data in commercial pig and poultry data, where we have data sets of more than 10,000 animals to work with. So to say a little bit more about extending genomic prediction models, how do we do that? Well, in regular genomic prediction, such as GBLUP, if we have M SNPs, we just assume that every SNP explains 1 over M times the genetic variant. But in the new models, we definitely want to relax this assumption using functional information as prior or additional information in the model. So to give you a bit of a flavor of some results we've obtained from using additional omics data, this is work from Bruno Perez on mice data that was that we've got access to by Gary Churchill from the Jackson Laboratory. And here what Bruno did, he looked at 10 different traits. I'm just showing results for 3 of them, being body weight at 10, 15, and 20 weeks of age. And here, these graphs, they show partitioning of the variance. So the first column here shows that for body weight at 10 weeks of age, 42% of the variance can be explained by SNP, so the heritability is 42%. That goes to 35 and 37% at 15 weeks of age. The second bar in each of the plots shows the proportion of variance explained if we consider only individual level gene expression data of these animals. And then the 3 plots show that actually with the gene expression data, we can explain almost twice as much of the phenotypic variance as using the SNPs. Then the third bar in every plot shows what if we put them together, and then we can actually see that the 2 of them together don't explain that much more variation than the gene expression data alone. So Bruno also in the same data evaluated the accuracy of predicting those body weight phenotypes at 10, 15, and 20 weeks of age. And the pattern here is very similar as for the partitioning of the variances. With the SNPs alone, we achieve accuracies around 0.2, but using the gene expression, we achieve accuracies of predicting phenotypes anywhere in the range from 0.5 to 0.6, so it's considerably higher. So just to summarize this part of the presentation is if we used individual level gene expression data in mice, we could actually explain considerably more phenotypic variance than using the SNP data alone. And it also considerably improved accuracy of predicting phenotypes. And what I didn't show is that, but for for most of the animals, gene expression was measured at different moments in time. And Bruno also found that if the gene expression is measured closer to the moment where the phenotype has been measured, that then both the explained variance and the prediction accuracy are also higher, which biologically makes sense, but it was nice to actually see this confirmed in this data. So, well, I'm sure there will be lots of related presentations during this year's EAP meeting. I'm just highlighting a few that either I'm involved with or are connected to the Gene Switch project. So the first 2 is a presentation by Bruno Perez about gradient boosting machine learning method for genomic prediction in the mice population, where I just show some results from. And funny, Molandin from France is presenting her extended Bayesian genomic prediction models. These presentations are both in session 46 on Wednesday, and another potentially interesting presentation is by Lisanne Verschuren on Thursday in session 66, where she took a multi-omics approach to predict feed efficiency in pigs. So next to SNPs, she also used microbiomic and metabolomic data, and she actually showed some results that are in agreement with those results on the mice data I just showed previously. So to summarize, impact of using other omics data, it gives us a better handle on phenotypic variance. It allows us to improve the accuracy of predicting phenotypes, but something we still haven't quite achieved yet is that it also helps to improve accuracy of predicting breeding values, and that's in the end, from the perspective of a breeding program, what we're after. Finally, the impact of using FAANG data. New models are being developed and GeneSwitch, but also other projects. There's quite a few people working on this. The FAANG data is currently being generated, and in the project GeneSwitch, we will demonstrate the potential of this in terms of impact on accuracy of genomic prediction in the next 1 or 2 years. So with that, I would like to thank you for your attention.